Awesome. Welcome, everybody. My name is Elise McCormick, and I am a PhD student in the Organismal and Evolutionary Biology program at UMass Amherst. And I'm going to be your MC for tonight's Life Science Cafe. Just to give a little bit of background about our group, Life Science Cafe was established in 2011 and is organized by graduate student researchers at the University of Massachusetts Amherst. Each month, we invite experts to share and discuss their research. And we would like to thank our funding sources, in particular, the Organismic and Evolutionary Biology program, the Graduate Student Senate, and graduate school at UMass, the National Science Foundation, and our very generous UMass Gives donors. If you'd like, you can subscribe to our mailing list and stay tuned for future cafes. As an additional note, hopefully it was no.
Let's try that. Hello. So we are now hosting hybrid events to benefit both our virtual and in-person viewers. And we're also trying to make our events more accessible, which includes closed captioning and events in other languages as well. So if there's anything that you feel would improve our accessibility of events, please feel free to reach out to either myself or one of the other students that are here and let us know about what your ideas might be. We would also greatly appreciate it if you would fill out both our in-person and virtual sign-in sheet. Our in-person sheet is kind of up there on the small lectern. And so this way we can continue to bring you cafes on the topics that you guys are interested in hearing. So without further ado, let's start our cell biology panel. So today I have the pleasure of introducing Dr. Marc Edwards, Dr. Katrina Velle, and Dr. Arif Ashraf for our cell biology panel. Thank you guys so much for joining us today. So as we get started, I don't know if you guys want to kind of go in a line and introduce yourself and just tell us a little bit about you.
Used to this one now. I am Marc Edwards. I am an assistant professor at Amherst College, but 100 feet that way, basically. I've been there for, this is my 5th year, I think. It's a kind of a blur. The COVID years, I don't know how to count them, but yeah, I've been there for about 5 years. I run a research lab there with now 8 very rambunctious undergraduate students. And I study cell migration and cell signaling in dictyostelium. And now, thanks in part to Katrina, I also think about osmoregulation in dictyostelium, how they regulate water content through their contractile vacuoles. But I'm a teacher, researcher, scholar, all of the above, and we'll get into it a little bit later, but that's kind of why I'm at Amherst, to think about both doing the research and the discovery, but also the training, but also thinking about how we welcome new cell biologists into the academy, how we get them interested and excited, but more on that later.
Hi, I'm Katrina Velle. I'm a postdoc in the Fritz Lehland lab at UMass Amherst. I've also been there for, I think, around 5 years. Yeah, the COVID years should blur together. And I study something called Naegleria, which is an amoeba that maybe you've never heard of. But if you have heard of it, you've probably heard of it in the context of the brain-eating amoeba. So this amoeba causes a deadly brain infection, and we don't have any really good, reliable treatments for it. So my work looks at the basic cell biology of Naegleria to try to look for weaknesses in it and things that we could potentially target with drugs to help battle this deadly infection. And some of the traits I'm interested in looking at are osmoregulation, which I hope to get into a little bit later, as well as cell motility, how these cells are crawling around, which is important for them to be able to infect your brain, as well as a little bit of cell division, which Arif is going to talk about next.
Thanks for inviting to this panel. I'm Arif Ashraf. I work as a postdoc with Michel Passer at the University of Massachusetts Amherst. I actually work with plant, and the way I'm connected and part of this panel because I work with plant, but I look at the cell. And the very fundamental things I look at the cell, all of us has not only one cell, many, many different types of cell, and they come from the cell division and produce a different cell types. So I'm very excited to be part of this panel and talk more about that.
Awesome. Thank you guys so much. So the first thing, I've got a question kind of for Katrina. What would you say makes up a cell?
Thank you for asking that question. I have a slide for this. It is slide number 7, please. Oh, thank you. It's already up there. All right. So we do have to start with this kind of little book reporty definition of what is a cell before we really dive in here. So I thought I would start with this really textbook example that many of you have probably seen at some point, whether it was middle school, high school, undergrad, where you have 3 cells here, an animal cell, a plant cell, and a bacterial cell, which is not all there is. We'll get there. And oh, these animations are gonna be annoying for you. So a cell is basically the smallest unit of a living thing. In some cases, like the amoeba I study, it's the whole thing. It's a single-celled organism. And it's basically something that's contained by a membrane. So it has a plasma membrane on the outside, sometimes a cell wall for plant cells. And then it also has genetic material on the inside. For many cell types, particularly the ones that the 3 of us study, we're looking at eukaryotic cells. So a eukaryotic cell has DNA contained with the nucleus, so another layer of membrane there. Then one more. And then there's a bunch of other stuff, which I think this diagram does a really good job of showing that there's a ton of other stuff in the cell. You have different organelles, you have mitochondria, which are making all the ATP that your cell needs, the energy it needs, as well as a cytoskeleton, which I think all 3 of us are very interested in, actin and microtubule filaments that give the cell structure and support, help move things around. But I do find this textbook diagram very boring. And if you look at real cells in real life, you can see they're really beautiful. So in this image, all of those different cells are set to the same 5-micron scale. So you can see over on the left, there's a plant cell, or many plant cells, a bunch of plant cells from the cassette lab. And then there's some amoeba in the middle, and that giant cell you see on the upper right is a mammalian cell. And if we can go to the next slide, please. Thank you. And when we think about how all of these things are related, we can kind of gain some evolutionary perspective. So if you were to not have been here for the past 5 minutes, and I told you to think of a cell type, I think many of you would probably be thinking of a mammalian cell, like a mouse cell, or maybe you go really out there and think of a fruit fly cell or a frog cell. But all of those things are really relatively related. In fact, we last shared a common ancestor with a sponge, which is very different than us, about 730 million years ago. In the grand scheme of things, that's not super long. We also, this group shares an ancestor with brewer's yeast about 1.3 billion years ago. So now we're starting to get more into deeper evolutionary history. And if you go even further out, around 1.6 billion years, that's how far you have to go back to find a common ancestor between dictyostelium cells that Marc studies and our cells. And then even more out there, we have the plant cells that Arif studies and the Naegleria that I study. And since these all diverged about 1.7 billion years ago, we can look for comparisons between these different lineages, and anything that's kind of the same between these groups could be something that existed in the last common eukaryotic cell. So we can kind of get this picture of what the first eukaryotic cells might have looked like, which I think is a really exciting perspective on this. That concludes this book report part.
Well, that was the best book report that I've ever seen. So I thought that was. So next question is kind of an open question for whoever would like to tackle it. But in your own words, why is studying these cells and their movements so important? Who wants it?
I'll get to important. Cool. I'll talk about this at the end, but cool. That's how I got to it. The first time looking at cells under the microscope, I remember looking at my high school teacher like, "Was this here all along? Where have you been hiding these things?" He told me to pick some, swish it on some pond water, put it on a slide, and look at it, and I was like, I was looking at National Geographic stuff of the same old lion chasing the same old gazelle, but like under the microscope is a world with matched complexity. And I think extrapolating back out from that to think about why it's important, I think we're explorers by nature. And I think exploring back to, as Katrina put it in her book report, back to the last common ancestors, the thought of reaching back so that we could reach forward, so that we learn to better think about diseases, how we work, how the diversity of life has evolved, why the world looks the way it looks around us, and where it might be headed and where we might be headed. I think are really cool questions whose origins can be traced all the way back to a single cell, and some would argue much further back than that. But the cell is a basic unit of structure, function, and complexity, and thinking about the evolution of that, I think, makes it both cool and again important as we, sort of fulfill a larger mission of exploring the world around us.
That's so interesting, and I love that perspective. That's just so inspiring. So I've got another question. This one is for you, Arif. So we're talking a lot about cell movement, but also cell division, and that's something that's part of the work that you do. If you could tell us a little bit more about that and what sort of cell polarity and division is, give us a little bit of context, that would be awesome.
Thank you. So actually, let's connect all of us to a very single cell. What is true that all of us were at some point one cell, and from here we came up to this point. And think about, you have the skin cells, you have kidney cells, and they do different functions. And if I have only the skin cells, that cannot do the function of the kidney. So I need cells of the various kinds, and they should do different kind of function. So what is very interesting that if you think about as an animal organism, we all need those cells. And how those cells are coming, so even 200 years back, we didn't know how the new cells come. So there was a kind of theory scientists used to propose that's called the cell theory, that every cell is coming from another cell. But no one didn't know, okay, I understand it's coming from another cell, but how? Then at some point, because of the great advancement of the microscope, we actually saw that the things are getting apart and creating two cells. So right now, although we know and understand the cell theory and cell division, but there are so many things we actually don't know. Like how a cell knows that when to divide, like where to divide. So one of the fundamental things when you think about, and going back to the question, how we create all these new cell types, it actually has to produce when it divides. If you keep producing the same kind of cell, I would have only one cell type. So I've actually point for this slide number 13, why it is relevant in a sense that when you think about the cells need to produce two different cell types, on that type, actually the nucleus that actually contains all genetic content cannot sit inside one part. So here you see a plant nuclear cell where the nucleus is like a ball, just keep moving. And if you go to the next slide, you'll see that the nucleus or this movement is actually very awesome, that it not only moves, it also rotates. And these things actually very fundamental. And we ask, why should I care? Because the positioning of this organelle, we talk about the cell moves, but the positioning of movement of this organelle is actually decides where the next cell division should happen. So we'll talk more about that as we go further.
That's awesome. Thank you so much. So this is our kind of last question for the first part of vignette 1, and then I'll kind of turn it over to the audience for a minute so they can ask some questions. But Marc, you mentioned that you study cell movement. Could you give an example of how cell movement affects kind of the everyday life of a normal person?
Sure. Can we go to slide, I think it's 18. Yeah, so can you play that movie down on the bottom? So you have a bunch of cells entering a little maze on the left. Is it playing? Oops. Okay, well, the movie plays all the way through. They're entering a maze on the left, and they have to solve this little problem to get to what we can appreciate on the right is a source of a particular attractant. So they're detecting a cue off to the right of the slide. So when we think about how and why cells move, I think they have places to go. Is why I put this slide up here. So where they move. And much like us, they're exploring around, they're looking for cues and trying to figure out where they need to head to. It turns out that the evolution of complexity, one of the really critical tools for that was for cells to pick up and start moving around, rearranging themselves. Arif mentioned coming from a single cell. Well, single cell, after a few rounds of division, you have a ball of cells that have no particular polarity. They don't have front or back, dorsal or ventral side. And after a few more rounds of division, something incredible happens. They pick up and they start moving into place. They start chasing signals down, they start developing, and you start patterning out into a whole organism. There's also for organisms that are much simpler, like the slime mold dictyostelium that I study, they have imperative, just like we have, to find food and shelter, to divide, to replicate. And all of those things are involved searching and exploring. So cell migration turns out to be a really fundamental cellular property. And not just the question of cells moving, but also the question of when cells stop moving and why they stop moving is also really important overall. But I think, yeah, cells move because they got places to go, which is usually how I start my lecture on motility at Amherst College, and thinking about why they're moving, where they're going, how they get there, how they're detecting things, and really how they execute on really complex behavior. I'll end with my dicty cells living in the soil, chase bacteria around relentlessly, but they don't like to get too close to the sun because it warms up. But they don't want to dive too deeply down into the soil because it cools down a little bit. They really hate fungus and they'll scatter at any sign of the fungus. Now, this is not a policeman or police person in the middle of traffic, directing traffic, saying, you stop, you go, you come, you go over there, slow down, what are you doing, buddy? Like, none of that. They don't have a brain or anything that complex that lives outside of them that gives them this direction. So they have to process all those signals. They have to integrate all those things. So I think that for me is the really another set of really side of the question, another set of really interesting things, and thinking about how cells are going, where they need to go, and just as soon as they determine which direction they want to.
Okay, so that brings us to the end of our first vignette. If there's anyone in the audience that's got a question for our panelists, now would be a great time.
How does Nigeria get inside the brain? I mean, does it have appetite only for brain cells or other kinds?
So if I can repeat that question for Zoom real fast. How does the brain eating amoeba find the brain, get into the brain, and all of that jazz?
Thanks for that question. It's all an accident. It doesn't want to be in your brain. It doesn't want to eat your brain, but there's no bacteria in your brain. So it's kind of like slim pickings. This is all I got to eat, so I'm going to eat it. So people typically get infected if they go swimming in a pond on a hot day and get water really far up their nose. So it's not just like a little water up your nose. It's like get a lot of water up your nose, or if you're on like a water slide in a water park that hasn't treated their water properly. But somehow the amoebae are introduced like far up into your sinuses. And then they can actually find these little channels that are in your skull, which is disturbing to know they're there. If you don't know that you have tiny holes in the base of your skull, you do. That's how your olfactory neurons get from your nose into your brain, so you can sense smell. And they follow those olfactory neurons and crawl along them to get into the brain. I think I have a slide for this, but I don't want to hog all the questions.
I would show it.
Yeah.
All right, great. So slide number 10. So I've been doing these experiments looking at how cells are interacting with these tiny little channels. So here, if you look at this movie, you can see the cells in that blue area are just crawling around freely. And then that green piece is a kind of like a piece of plastic that has these tiny holes in them. And you can see the amoebae are starting to line up along the edge and they're probing into these channels. And once they make contact, they don't turn around and crawl the other way. They just kind of keep probing until they get in. And then if you go to the next slide, once they do get into these channels, they move really quickly and they don't turn around. So I think, so here we go. The top video shows a cell crawling through a channel. The bottom video shows a cell whose path has been blocked by a little piece of something. So you can see they don't turn around unless they come across a complete blockage. They just kind of keep going. So it's really disturbing to think about in an infection context where the amoebae might be finding these little holes that they can get into. There's something about that interaction is favorable, so they continue to interact with them and keep crawling in one direction until they get into the brain. And for another fun video, the next slide has some constriction points. So you can see that these amoebae are actually really great at even getting through these really narrow constriction points. So as long as the path isn't completely blocked, they can make it in. But again, they don't want to be in your brain. It's an accident. But something about them drives them into these narrow spaces. Oh, nose plugs. This is the PSA. Wear nose plugs.
Nose plugs for sure. Now I will never be without them. That's awesome. That's awesome. Does anybody else have any questions for our panelists? Yeah.
So the principle, you mentioned how those different sensories those used to know. I think it was like 2 weeks ago, we had a signal speaker come for our program talking about how some signals can override cell division. Have you noticed in the organism that you open, is there one particular signal that overrides all of those?
Okay, so I'm gonna try to summarize that question for Zoom really quick. And tell me if I got it wrong, but have you noticed in your work that there's one particular cell signal that overrides all the other cell signals? Okay.
I've been batting this answer around in my head for the last 5 seconds. Because it's a little morbid, but death. When it's time for cells to go, they're going. And whatever else they're doing, they're going to stop. That is, and eukaryotic cells do this. So the ones that are further along the tree do this in really highly prescriptive ways. But Dicty has some as well. Dictyostelium, in one example, this Dictyostelium has a multiphasic, biphasic life cycle. When there's food aplenty, it lives in this sort of cherubic single cell stage. And I think slide, let's see, maybe slide 21. So this is a picture of what Dicty looks like when there's a lot of food around. So we use fluorescent proteins to mark individual fluorescent proteins as fusions to mark individual proteins, so we can actually see their distribution. And I study the mechanisms that allow cells to move, and the mechanisms that allow them to move also allow them to feed. So those little cup-shaped projections that you're seeing there are the outcroppings of the membrane that Dicty uses to swallow up membrane and take stuff inside. And they also use similar mechanisms to move. When Dicty runs out of food here, it starts developing. So it changes a bunch of the pattern of gene expressions, and it enters into this multicellular phase. It promises it's going somewhere. And they'll make a little ball of cells, they'll aggregate together, they'll crawl out into a little slug. And after they crawl into a little slug, they make a little fruiting body that looks very much like a mushroom stalk. The cells in that stalk sacrifice themselves for the greater good of the Dicty. And the signals that trigger them to make that sacrifice, those types of signals are very hard to override. And I think is sort of the perfect answer to that question, albeit a little bit morbid. Other things, I used to do this a little bit when I was a postdoc, when there are multiple different signals that will lead Dicty to move. They will migrate towards bacteria, they'll migrate away from some fungi. They will migrate to food and sugar sources if they have to. They can actually align themselves along external electric gradients and external electric fields, so they can sense that as well. They will migrate to touch and pressure. And at one point, I had a dream as a postdoc, as one often does as a postdoc. The PIs in the audience are smiling. Oh, that's an inside joke. Okay. I wanted to pit all of the different signals against each other in one massive experiment, all done at the same time. My boss was like, that is insane. Do not do this. But of course, I stayed up many a late night and I built my Frankenstein contraption. I set up an electric field. I used a microcapillary glass needle filled with an attractant. And I pit all of these things against each other. And then I squashed the Dictee as well, and in a very fine chamber. And I did all of this, and I found absolutely nothing, which is very it didn't teach me anything other than I could build Mary Shelley's monster in a dish. But it was really interesting to see them in real time fight to integrate all of those different signals. And I think if anyone won, it was confinement. They would follow a direction pretty randomly in confinement, and they would start ignoring other signals. But so for moving cells, as the owner of that tiny bit of cell motility trivia, because I did this silly experiment, confinement, but overall, when it's time for cells to go, they're going.
All right, so thank you guys so much. Now we're going to move on into vignette 2. So I think you guys have set up a very inspiring and exciting stage for cell biology right now. But if each of you could kind of go into just a little bit more detail about your actual study system, and talk a little bit about what questions you're trying to answer with that study system.
So I little bit mentioned that I work with plant, and so you may think that what kind of plant, what kind of cells I look at. So which is a very basic thing. We all know that plants go through photosynthesis. But for that, actually, they need to take out the carbon dioxide and release the oxygen. And they need some sort of pore that actually facilitates. So those pore are known as stomata. So those are also cell, but they can form a shape that can facilitate like a door to gas or a gas exchange. So, but the thing is that they don't look like this when they born at the first time. So we call that very early stage of the development, like our single cell, how we started. So on that case, what I do, that I watch them when they have a new cell type, new function. So for that case, I actually need to go very early of the stage where cell knows what to do. And what they need to know that I have to divide and I have to produce different cell types. So during that time, I'll actually indicate the slide number 16. So during the time, initial play, please. So during that time, we'll see that all these green things are moving around. There's a microtubule, one of the cytoskeleton moving things. And all these small balls are actually nucleus, which contains all the DNA. And as you can see, when they'll divide at the bottom, that they actually produce the cells, which doesn't look like same. And by not looking like same, they're actually doing the different function. And eventually, they're going to be those cells which actually facilitate those gas exchange for plant. So if you ask me why they're so important, they're important because if we miss that development stage, they'll produce the cells which cannot facilitate the gas exchange for plant. They cannot do that for the synthesis eventually. But it may hard for you if you're not a plant biologist, why the cell division itself is very important. So I always give that example of like when you have cataract in our eye, we cannot see things, right? So that is actually the lens cell, which is transparent. But if that cell cannot divide properly, it will never become the transparent lens cell. It will become something else. So that's why with cataract, we cannot see. So if you go very deep down, you find why the cell division is very important and connected to not on our life plan and across the life system.
All right, so I've already told you a little bit about the brain-eating amoeba and might have drawn your attention to that aspect of it. But here I also just want to point out that I'm not actually working on that amoeba. I study a non-pathogenic species that's a relative of it, so it's very safe to work on in lab. And I'm looking at things like how these cells crawl, which I went into a little bit before. The first paper that came out of my postdoc, maybe slide, how about slide 9? Sure, thank you. The first real project I was working on as a postdoc was defining how these cells crawl. So here's a Naegleria cell crawling, if the video will agree to play.
It's playing her.
Imagine that it's crawling. So my first project was looking at how they're crawling. And what I found was that these cells, even though they diverged from animal cells, 1.7 billion years ago, they're relying on the same basic mechanisms. They're relying on the same actin network, that cytoskeletal network I talked about earlier, in order to drive their motility. And that's something that we think could be conserved then. It might have been an ancestral trait present in the last common eukaryotic ancestor, is moving using this actin cytoskeleton and some specific upstream proteins to polymerize actin. However, how cells crawl when they're on a flat glass cover slip is not how they're going to be crawling when they're out in nature or when they're in an infection scenario. And as Mark pointed out before, when you put cells under compression or when they're in different environments, that really changes how the cell moves. So right now, my research is really focused on looking at how these cells are crawling when they're in different environments, like in these microchannels or when they're kind of squished between two cover slips. And I'm hoping to look at them in kind of more complex environments, like crawling through different networks and gels, to see what changes about their motility there. Yeah, so cell migration is where I'm going.
Could you go to slide 19? Okay, so this is what we do in my lab.
I don't know where to move the factions.
That's fine.
Over here.
Yeah, like, that's okay. That's okay. So I got really interested as a grad student and a postdoc with just moving cells. I really needed to figure out, I guess, since I was a high school student, how cells move. They were crawling around. I thought it was fascinating. I didn't think anything that small could move, let alone move deliberately or with intentions to a particular source or what have you. So I got in my lab to thinking about that all of these cellular processes, cell migration, movement, division, are reliant in principle on molecules colliding with each other with sort of the right orientation, right energy to carry out chemical reactions at their root, right? So we have proteins interacting with each other that enable all cellular function. So the web of interactions, then the nature of those interactions, how, when, where proteins interact with each other becomes really critical for understanding how cells are able to do the complex things they do. So I start with standing on the shoulders of lots of scientists who figured out that web of interactions in the top left corner. And then I think about cellular behavior under the microscope, and I think the picture in the top left is really static, right? It does not reflect the dynamic nature of the interactions within cells. There are no feedback loops. There is no sense of the massive amount of promiscuity that's in cells. It doesn't give you a picture as to why cells are so robust to mutation, to perturbation. So what I really try to do is to give life to the diagram up on the top left by exploring how cells move under a microscope. So I make the same perturbations that you might see in the top left and think about how that changes cell movement in different contexts. We use a little bit of computational biology. We like to model some of these interactions, because where we would like to get to is a place where we understand the molecular architecture of the networks of interactions that control cell movement, that we are able to manipulate those interactions to our own ends to make cells move in predictable ways. And I stole my own thunder. Really, I'd like to be able to predict how cells would move if we are at the helm and manning certain manipulations. I once had sort of an unfortunate run-in with funding agents of DARPA, the defense agency, where they're like, how much faster can you make the cells move? I'm like, well, I don't really care. If you want me to, so that I can stay funded, I'll pretend. And we got them to move about 2.5 times faster, which in biology world is amazing. I mean, we were ecstatic. I went back to the DARPA guy and he's like, really? 2.5 times? I'm like, yes, it's amazing. You have no idea how many long nights I had to stay up to figure out which of those levers to pull and which of those toggles to twist to get cells to do this. Because cells, after 1.8 billion years, pretty optimized. They're pretty settled in what they do and how they do it. But it is sort of an amazing journey to take to try to understand and then be able to manipulate and predict how exactly cells move. I'd love to be able to give a talk where this is how they start. These are the series of interactions that we think are principally important. And if you know this and you know enough of this, then you too can be able to manipulate and predict how cells move. Still, 2.5 times faster is plenty fast.
So Mark, I want to kind of touch on that briefly and come back to it a little bit. So you talked a lot about cell movement. How do cells know when to move?
My dictee don't seem to need much help in a dish. They just start going. But if I give them slide 20, play the movie on the left. Right. So what I've done here, and well, in both of these movies, you can kind of see it, is I have a glass capillary needle that I've pulled really, really thin, and I fill the capillary with an attractant, a chemoattractant, so a chemical that stimulates taxis or movement. And the dictee move pretty robustly to that needle source. And it turns out that to take dictee from this single cell phase to the multicellular phase, one of the first steps is a massive aggregation phase where they have to all find each other. They have to quorum sense, so they have to figure out how many neighbors they have around. Do they have a critical mass of neighbors? Are the neighbors all starving? They release a chemical to organize this concerted movement. And very often in cells, there are signals like this that trigger cells to move. The signals of a chemical nature. Katrina has given you not necessarily a signal, but certainly something that they sense that changes how they move, that influences where they move. So basic imperatives: food, finding security, getting away from fungus that might want to eat them, or larger things that might want to eat them, all inspire cellular movement. I'm also interested in things that we spent quite a lot of time in my field thinking about what makes cells move, and very little time thinking about what makes cells stop moving, which I think is something we should probably get to, because what we know is that when cells that have stopped moving or shouldn't be moving around start moving again, we get in a lot of trouble in multicellular systems. That's at the root of metastatic cancer. It's certainly a lot of defects that are congenital disorders. But yeah, they'll typically explore around pretty well on their own, but you can definitely give them cause and reason, like we're seeing in the movies here, and that can be translated into a number of natural scenarios.
Very, very cool. So this question is for Katrina. Your research focuses a little bit more now on cell migration and also division and osmoregulation. And we touched a little bit on osmoregulation earlier, but we haven't actually talked about what it is. Could you define that for us and then tell us a little bit about your research on it?
Yeah, thanks for that question. So I have a couple of slides starting on 23. Oh, you're already there. All right, so osmoregulation is something you're probably not constantly thinking about, but I mean, your kidneys are kind of taking care of it for you. And if you're a single-celled organism, and particularly one that lacks a cell wall, and you live in a pond, osmosis is going to be a problem. So, can you click? Thank you. Osmosis is going to drive water across the cell membrane and into the cell. And if the cell doesn't do anything about this, it will continue to swell until it eventually bursts. So to get around this, many eukaryotes, including dictee and Naegleria, use a specialized organelle called a contractile vacuole. So that way, when water enters the cell, it gets collected into this contractile vacuole network, which will then become this kind of full bladder that will periodically make contact with the plasma membrane, with that cell surface, and then it will collapse. And that collapsing will drive the water out of the cell. And, you know, I'm sure you love these low budget animations, but this looks really amazing in actual cells when you look at them under a microscope. So here are some different examples. The one on the left is Chlamydomonas, which is a plant. It's a unicellular green algae. And you can see it has these kind of two circles at the base of the flagella that kind of grow and shrink on these off beats. So those are what's squeezing out the water there. Paramecium, which is that second example, has these canals that extend into the cytosol and collect the excess water there. And then those drain into this central bladder in the middle that you can see then pumps the water out of the cell. And then we have different amoebae, like acanthamoeba, and then Dictyostelium, the example on the right. That cell came from Mark. Thank you, Mark. And that's actually how we started to collaborate with each other. We're actually working on a paper right now about these contractile vacuoles in both Naegleria and Dicty. So I have one more slide just showing you what they look like in Naegleria. And here I've color coded them so that once the bladder is full, it turns a color and then you can watch it empty. So there's one pump happening, and then you can see the water being collected. And those smaller vacuoles merging to make a bladder for the next pump, and it pumps that out of the cell. So I think looking at these organelles, it's hard not to become fascinated with them and want to know like what makes them tick, how are they doing this, how are they pumping water out of the cell. And this is also a really critical process for all cells because if you're not controlling your osmolarity, then you're going to swell and burst, which also makes this a great drug target. Just to come back to that idea of developing drug targets to tackle Naegleria infection.
Thank you so much. Okay, so this is our last question of vignette 2, and this one's for Arif. So your work has kind of talked a lot about the nucleus, as you mentioned earlier, and showing that the nucleus decides whether the cell is going to divide. Could you tell us a little bit more about that?
I really need to show the movie that it makes sense, like slide 16. Yeah, I can start talking. And then so yeah, so let's say imagine this room as a cell, and then the nucleus is a kind of center of the cell sitting in the middle. But we need to divide this room into an unequal fashion. So in that case, all my genetic material, if it sits in the middle of the room, I cannot do that unequal distribution of the room. And cells, as this room knows that, okay, I have to move to that direction. So the nucleus itself, with all the genetic material, it moves towards my right. But the question is how cell knows, that the nucleus knows that I have to go to this direction. The cell has those compass, like some of this protein. They'll sit there and tell, okay, this is the direction. It's like a Google map for the cell. And once it happens, the nucleus, all the DNA and genetic material moves towards the side of the cell of the room, and it divides. Only that time they will produce the cell, which doesn't look like equally volumetric or same or totally different type. So that's why if I am a nucleus and I'm sitting in the middle of the cell, but I have to divide unequally, I cannot sit here. But also it's a long-term question, like, do I decide as a nucleus and the cell follow me, or the cell decides and then I follow that? So basically my research tried to tackle this very fundamental question. And what I can tell you so far, what we discovered, that the nucleus moves, and wherever the nucleus is, cell follows that and divides there. So which is actually kind of answer the fundamental questions in the biology. So, and I hope that this will be part of the bigger knowledge in the long run and part of the textbook someday.
That is so interesting. I love the idea of cells having Google Maps. That's just such a fun analogy. Okay, so this is the end of vignette 2, and I'd like to take a chance to talk to our audience. Do you guys have any questions for our panelists? Yeah.
This might be going too geopolitical, but we already know humans will attack other humans, unfortunately. Are there examples of cells, identical cells, attacking their own kind?
Okay, so let me summarize that for Zoom really quick. And the question is, are there cells that will attack other cells of the same type? Whoever wants it.
Try it. So the first example really coming to my mind are our immune cells. So I'm not an immunologist, but there are immune cells that can detect cancer cells and will attack them. So I think that's the closest thing I can think of. Naegleria cells won't eat each other if they're alive, but if a Naegleria cell dies, the other cells will eat it. So they eat their dead friends, which is interesting. Yeah, that's what I have for that. I don't know if anyone has anything to add or a better example.
I don't have that. I don't have that many great examples like similar cell types go for each other, but usually the cell types that target other kind of organism to steal their like say, chloroplast or nucleus. So they are like stealing kind of examples, but not. I don't familiar with the other kind of examples.
We've given some of those cells fun names. There are cytotoxic lymphocytes. There are natural killer cells. What else, boy? Macrophages, although that doesn't really sound that scary. Dicty will nibble. After this, I'll show you a movie. I think it's on my phone that I show my students all the time of Dicty nibbling on another Dicty while they're alive. And they usually spit the Dicty back out. And the Dicty that gets spat out usually gets very angry and moves away really quickly. I think it's a real thing. When you stare at cells for as long as I have, you start to make up stories for them. But I do think, like much like Naegleria, they won't really eat live Dicty, won't eat other live Dicty. But if they're dead and if they're confused, they will take a little bit of a nibble.
Any other questions from our audience? Yes.
This is for any of you, but is there one mystery that you'd like to solve before you retire?
All right, so for Zoom,
What's the one mystery?
is there one mystery you'd like to solve before you retire? Who wants it? Or any?
I can quickly start. Actually, there are many of those questions you want to answer. So this is still a tough question just to pick one. But if I pick something right now, I would pick that one question like when a cell knows the direction and nucleus wants to follow that, we really don't know how they communicate. So it sounds like very obvious, but how they communicate with that polar protein to go to that. We kind of know that nucleus use the cytoskeleton track, like the train track we follow, but how that train track or that direction actually connect. That would be kind of one of the top question I want to answer. And it is kind of not only answering the plant, it will answer in any biological system, basically.
This is not my area of specialty at all. Post tenure, I'm gonna try to figure this out. Dicty farm. It turns out that Dicty can swallow but not destroy bacteria. They farm them, so the spores that form in that little fruiting body, the fruit body blows away, new Dicty germinates, and they start that life cycle over again. They farm bacteria, which when I found out, I was like, what? And there are trade-offs. There's trade-offs between the fitness, so how well the spores do when they have too many farmers. They know which bacteria to farm and which bacteria they really don't want to farm. There are other cells called sentinel cells that fight off infections by bacteria that they really don't want to keep. And there is a whole world there right at the precipice between single cells and multicellularity, which is itself its own really interesting field. But I'd really love to figure out where that came from, where the point in the decision tree was, and where Dicty sits relative to that point. Like when did it pick up farming? When did it pick up altruism? When did it pick up so there are some cells that sacrifice themselves to make the stalk, and so that other cells can make the fruiting body so that the whole species can actually survive. There are a lot of what seems like conscious decisions being made here that I think we can trace the evolutionary origins of, but farming is the one that I am totally fascinated by. It's an example of when cells stop moving. The farmers and the sentinels have really different motility, and we really don't understand. The sentinels tend to get left behind in the wake of the moving Dicty slug before it actually sits down and makes its fruiting body. And I just think all of that is really fascinating. Thinking about a single cell that has all of the information and all of the right moves to enter into this multicellular phase, that's not just structural, that they build this towering structure and go disposably, but they are optimizing. They are trading off between farming and sentinels and killing. And I have to learn a lot of biology to be able to do this, but that's why I became a scientist. When I finish this little bit that I'm doing here, I'm gonna be, I'm gonna write a book about dicty farming, just you.
Yeah.
I'm also excited for that book. Okay, so we're gonna start our third vignette now. And I want to start off with, this is something that if you could all touch on this, that would be great. What is the thing that you love most about your profession?
So I really like looking at cells on a microscope. And I kind of can't believe that I found a job where I get paid to look at cells on a microscope. Like Mark, what you were saying about when you were in high school and looked at pond water and just like it blew your mind, like that still happens like every time I go on the microscope and look at cells moving and see something new. Also with Naegleria, not many people study it, so we don't have a ton of information about it. So anytime I sit down at the microscope, there's an opportunity there to see something for the first time and to like make a discovery that no one has ever noticed before. So I find that aspect really thrilling.
I'll agree with Katrina in many ways. So sometimes when I see things, the feeling I have that only I am seeing those things. And when I go home, sometimes I feel like my life is really valuable because I need to pass that information at least to my boss, to my lab mate. If something happens to me, this knowledge will not be lost. So otherwise, when I go back home, I don't find it much meaning. But those things I know and I see and observe, it gives really the meaning of life, my life. So I always say that science is the reason I believe in love, and microscopy is actually helping in that.
Could you go to slide 28? So I spent about 8 months of my life trying to make that microscope contraction on the right work to image through volumes of cells. It never really worked. It produced this one movie here for me. You can play that one. Okay, maybe. But I stared at this movie. I played the movie for my boss, and the movie, I never made it pretty. I never exported it in the way that we were really supposed to. I always play it just like this because you can't hear it, but the audio in the background is of me talking to my boss, and it's like, you see that little squiggly right there? That's the thing I've been talking about. And he's looking back at me like, I don't see the squiggly, and maybe we should away from this microscope. It's like, no, the squiggly is there. It is real. I'm telling you, see that little center spot in the middle of that little angry face thingy? Right where that spot is? That was my squiggly. It's like, that is a thing. There's a thing at the back, at the base of the structure that's different from everything else. My boss is like, I don't want to say it. But it became a thing. It 2 papers worth and a whole lab later, it's a real thing. And I love this, that's half of what I love. And that's the ability to watch cells do their thing, to appreciate how much led up to the moment of me sitting at a microscope, looking at a cell doing a thing, seeing a squiggly in the side of a thing. It's my squiggly and it worked. That's the name of my memoir, my squiggly. But the other half, and when you become a PI, a principal investigator, you don't really see. I'm like turning it around. I was like, look, Peter, that thing at the back, see that little bright dot? That's my squiggly. Didn't believe it, but it's true. Anyway, the other thing that I like is that it just happened to me just a week ago. My boss and I switched places. I am now the PI. My student sat me down, my 19-year-old undergraduate student, and he showed me his squiggle. He was like, look, I think I see something here, and it looks like this. And for a second, I was like, no, go away. There's no respect. I'm like, oh God, no, I am my boss. But I love being there to see them find their squigglies. I love being there at the moment that they fall in love with it like I did. I am a professor. I think maybe this question was going to come to me eventually, but I'm a professor because I feel indebted to my school teacher, to my undergraduate mentor, my PhD mentor, who let me explore, let me spend, 10, 15, 20 years of my life actively looking, chasing my own squiggly, finding it, you know, like really, yeah, exactly. I was really trying, Rachel. I was really trying. But yeah, I love being there at the moment. I love being able to partake and share in that moment of, you know, students finding a little bit of joy like I did when I was a kid in the science and in microscopy. I love being able to be there to shape laboratories, to shape undergraduate education, to think about who gets to be a scientist, who gets to have this experience, how we make that available to the broadest cross-section of people, coming from the broadest cross-section of places. So it's the science, it's looking at things under microscopes, absolutely. But now increasingly it's in providing students the opportunities to do the same.
So it's kind of a building question on that. What were each of your paths to becoming cell biologists? How did you get hooked?
I can start with that. Actually, I didn't like biology. I didn't read much biology when I was in high school because my dad wanted me to be a civil engineer like him. So I like math. But what happened that I used to tell him that I want to make movie. Whatever I study, I will make some film. That was my goal. And over the time, I realized that I would watch a lot of weird movies. No one watches those. And then I went to the college, like university, and that is the time I actually looked into the biology and I started reading. My grades are not great, but I love those books and the color illustrations. And over the time, I moved on and becoming from the plant, look at the plant cell and becoming more into the cells. And when I was in my PhD, I'm actually originally from Bangladesh and I was in Japan. I went back at home to see my dad because he was very sick. And he was asking me, okay, explain me what you do because I don't understand what you do. So I said, look, I look at how the proteins move inside the cell, like you move, go to the office, go to the supermarket, all these sort of things. If there's some rain, you will not go and skip some space or something like that. So he said that, oh, it really looks like your success depends on if you see something which other people cannot see. I said, yes, exactly. So he was telling me that I always hope you see things which other people never seen before. So that was kind of the best wishes for me. And at the same time, when last time I saw him and I was leaving the country, so he was asking me, did you remember that you always wanted to make movies? You wanted to become a filmmaker? So I said, yes, now I remember. And I know how stupid that idea was because it is not a real job actually in so many sense. So that was the last sentence he told me. He said, no matter what, if you want to make movie, you will make movie in one way or another. And basically I left and it's been 6 years. I couldn't go back home for many reasons and he passed away. And when I make all these movies, every time for lab meeting, for presentation, I feel like exactly, I'm making movies, I'm becoming a filmmaker and I'm making the film of life. Nothing can beat that with anything. So I felt like that I became a scientist through a very philosophical journey. And I think a lot of these things are this way. So I felt that I'm very lucky that instead of looking at the telescope, I walk to the microscope and I don't have to move and go anywhere, which I really don't like to go anywhere. This is that kind of fits with my personality and everything. So this was kind of a funny and sad journey for me to become a self-biologist.
Yeah, my path was pretty boring and direct. I liked biology when I was in high school. So when I got to college, I was like, let's try this. The one thing that's not boring about this journey is I hated it initially. And I quit biology, became an art major, transferred to a different school for their art program, spent 2 semesters as an art major, really loved it, but I didn't have the passion for it. I noticed that everyone else in my classes did. They were doodling during lectures. I was like, I don't have this level of commitment. So I decided to give biology another try, and the second time around, I really loved it. So from there, I actually made it to like really late, like maybe November or December of my senior year before I was like, oh, I've got to do something after. And then I panicked and applied to grad schools. And I'm glad that I panicked and applied to grad schools. And I'm even gladder that I made it into one. And then in grad school, the first time I looked at cells on a microscope, I was hooked and I was like, let's find a way to do this for the rest of my life.
Yeah, I was 11. I was sitting in the back of my high school teacher's class. I was I thought surreptitiously drinking a milkshake through a hole at a board in the desk. And my high school teacher just walked up to me. Well, he called me, he's Edwards, bring the milkshake, the strawberry milkshake. I was like, I didn't know. Anyway, he put the milkshake on the table. He broke down the different components of the milkshake, the enzymes in the body that would deal with them, how they'd be absorbed. And I was like, that is the most incredible thing I've ever heard. He's like, great. So you will write an essay of approximately 10,000 words. Cutting holes in the desk. I wrote the essay. I took it up to him and I was like, I just spent 8 hours with the Encyclopedia Britannica, like learning all of this stuff. This is the most incredible thing. He's like, yeah. Grabs the essay, throws it away. I was like, no, like I'm inspired. He's like, sure. So I kept going back to him. I was 11, so I wouldn't take like it was all integrated science at that point. It wasn't broken down in subjects yet. And he's like, okay. If you really want me to like teach you biology or let you into my class early, you have to clean all of the microscopes. I cleaned all the microscopes and I was like, this is the most incredible thing. And he's like, I really hope that would chase you away. So as a reward, he told me to go swish a little Q-tip in pond water. I put it on the side and I was 12 by that time. At this went on for a long time. And I just remember looking up to him one day and I was like, how do you do this for the rest of your life? Like, what is a person that studies, that looks at microscope, or what are they called? I don't know. Is it a doctor? So I thought I wanted to be a doctor. I went to undergrad and I'm like, oh, there are people who just do this for a living. They look at things under microscopes, and microscopes get much fancier than the rickety old compound scopes that we had in high school. And I never wavered. I was 11, 12 years old. I thought I wanted to be a biologist or scientist of some sort. I didn't have the language back then, but I think that single moment, I still remember what it felt like, and I can still get back to that moment where I was like, it's this. I don't want to be a fireman. I don't want to be a lawyer or whatever. Those noble professions all, but I need to be looking under microscopes. I need to be watching cells crawl, and that love has never left me. It's kind of straightforward for me in that way, but yeah, that was it.
I love those answers because I think it really highlights the diversity of what brings scientists all to the table and creates such important differences in our perspective. So that's so interesting. Speaking of kind of moving on to what's inspiring, what would you each tell anyone that is an aspiring scientist?
Yeah, I'll tell 2 stories. So number one, like as a high school student, I used to watch a lot of math videos. And there was an Oxford professor, Marc Edwards. He's very good in public speaking. So I used to see his documentaries. So he went to one of the Oxford local high school and he was telling the kids, okay, these are the hardest math problem in the century. So if you can solve any of these, that play mathematical association is going to give you $1 million. So all the students were very excited. And he said, if you think this is exciting, this is not the exciting part. Exciting part is coming next. If you solve any of this problem, your name will be written in the history even when you're gone. So I felt very hooked. I thought that if I can solve some problem, maybe I really want that feeling that when I'm gone, these things will stay there. But I really didn't have idea how it feels like to become a scientist. But one of my maternal uncle, he was a professor and he used to work with optimization and also things. So he said that you want to be a scientist, why? So I said, oh, it seems like very cool. And this is the documentary it was. So he said that he set me down and he said, you know, becoming a scientist is like coal mining business. So I said, what does it mean? He said that every morning you'll go to the tunnel and mining the coal. And when you come back home, sit on the dinner table, no matter how many times you wash, there will be some coal stain all over your body. And again, you go back. So it didn't really make sense, but he said that I come back home, but all my thoughts are in my mind. So I cannot sometimes communicate with people. Do you want that life? So I said, okay, I'll try and figure out. But now I see what he meant. And one of the biggest inspiration I should tell for the next generation scientists that maybe all of us would not get the Nobel Prize because it's limited. But if you do good science, that your discovery will be part of the textbook. So sometimes my sister asked, can you tell what you do? So I said, look, if I can do my job very well, your kids are going to read some of my discovery. So that is all I want to say. That is the biggest inspiration. And I always try to transfer that inspiration if some undergrad or someone working with me.
I'm not sure I have a great answer for this, but I think like the general advice I would give to a young, maybe wannabe scientist is that if you like what you're doing, keep doing it. And if you don't like what you're doing, change it. I mean, that's how I initially left biology and also how I came back to biology. And that's how I'm planning my career is just I'm going to keep at the things that I like.
I'm gonna echo that. I tell students all the time, it's like, look, you're 18, just do what you love, right? Just study what you love. Pick the classes that you think are interesting. You have an advisor. We won't let you like leave this place without a major. It won't happen. Just do what you love. And if it happens that science is a thing that you find interesting, read, explore all the opportunities to access it any way you can, but do what you love. Sort of follow your nose here. And if it leads you down the path of science, fantastic. If it leads the other way, it's okay. But I think, especially teaching undergraduate right now, I put a huge premium on making sure that they feel confident and comfortable and excited about exploring their own interests.
I think those are all really awesome perspectives. So this is our last question for vignette 3. And as people may be aware, February is Black History Month, and we're nearing the end of Black History Month. And I'd like to ask you guys if there's a particular Black cell biologist that we should be lifting up in kind of our understanding of cell biology and their contributions to the field.
Yeah, first of all, I really appreciate this is part of the discussion. I'll be very honest, there are not many Black plant cell biologists, but I want to highlight Tessa Smith. So her work is actually very fundamental to understand something called plasmodesmata. So let me break down that term. How do cells communicate? They have a junction, they have a tunnel, and actually that tunnel you are going to see on the image. And I'm really grateful that in our generation, we found some scientists like Tessa, who is helping us to go forward and understand the biology and cell better.
All right, I think the next, thank you. So I wanted to highlight, in honor of Black History Month, Dr. Roger Arliner Young, who was actually born in 1899 and died in the 1960s. So obviously a really, really tough time to be a Black woman and a Black woman in science. And I think she really deserves to be in textbooks, but I hadn't heard of her until I went to Woods Hole this past summer at the Marine Biological Institute, which is actually where she spent her summers for a while. And actually this picture of her was taken over in Woods Hole, Massachusetts. She became the first Black woman to publish a paper in the journal Science. And that paper was actually about contractile vacuoles in paramecium, which is why I added a little video of paramecium there. She was also the first Black woman to earn a doctorate in zoology in 1940. And it's tempting to stop there and let this be kind of like this uplifting story of, you know, a woman who battled against all odds and faced adversity and succeeded. But unfortunately, it's really a story of the system just completely and utterly failing her. So as you can imagine, she had a really tough time getting that doctorate. Her first advisor loaded a ton of extra work on her. So she was doing some administrative duties. She was scheduling courses for other professors. She had a large teaching load. She really had no time to do her own research. She actually failed her qualifying exam when she was at the University of Chicago, which might have had a lot to do with the fact that her advisor was an advocate for eugenics. So he was just constantly telling her she wasn't good enough and that she was mentally unfit to be a scientist. She eventually moved to the University of Pennsylvania and she did earn her doctorate there. But even then, even with a doctorate, she was unable to get a position that allowed her to start up a research lab. She didn't have the space or the funding to do so. So she was largely a teaching professor. But this did allow her the opportunity to engage in some civil rights work. So she did some really important work for different labor unions and for the NAACP. But again, unfortunately, that got her blacklisted from academia, and she was unable to secure another academic job after that. So academia really worked to push her out. And I think in a lot of ways, academia is still working to push people out, which is why I think events like this and doing things like highlighting and uplifting the voices of Black scientists is a great way to increase accessibility and really promote diversity in science that we so desperately need.
Yeah, I was really lucky when I went to Hopkins as a postdoc that I ran into maybe 5 or 6 other Black postdocs there, and we took really good care of each other. We all became professors, a couple of them at Hopkins and so forth. And I guess the person I'm highlighting is Samantha Lewis, who's a new professor, assistant professor at Berkeley, who studies the inheritance of mitochondrial DNA, so mitochondria that make all the ATP that Katrina referenced earlier, that really power a lot of the movements and activities in cells, have their own DNA, they have their own genome. And inheriting that with really high fidelity is really critical. There are a number of diseases that can be traced all the way back to defects in the inheritance of mitochondrial DNA. But I think she runs a really fantastic lab, Sam Lewis, lewislab.org, out of Berkeley. I love the way she does science. But I highlight her as she wasn't part of my cohort, but there's a really exciting cohort and a real number of really exciting young Black scientists popping up here, there, and everywhere. I think academia does work really hard to push us out still. It has been doing some things to make sure that folks are getting access. The efforts aren't perfect. There's still a really long way to go, but I put Sam Lewis's lab up there to highlight that there's a core of really exciting young cell biologists, developmental biologists coming through or landing jobs or are training amazing students who have really cool labs. So I think I live in the sort of Black scientist myself. It's easy to live on the other side, the pessimism of where things are, what the numbers are, why it hasn't moved faster. But I of late, I think I've landed on the other side, just being really excited for my friends, the folks that I came up with, the folks that I trained, the folks that I hear about, the folks that I see on Twitter popping up. Like, I have a new lab, like I'll always retweet them. Like I've even found a couple of scientists from where I'm from, Trinidad and Tobago. I'll see them popping up. I'm like, it just feels cool. It is inspiring. It is a time to reflect on I think the beauty of those experiences, those moments, and live in the optimism of the fact that there are quite a few young scientists, and that I think it's a little bit easier for us now, certainly than it was for Dr. Young, the story Katrina just told. There's reasons to hope, there's reasons to continue highlighting, continue uplifting, continue focusing on this. The problems we are trying to solve, the mysteries we are trying to unravel will require the full scope of our talent. And that requires and necessitates that we support Black science and Black scientists. That's what it means for talent to be sort of normally distributed across the world. That's what it means to try to solve, to do the unthinkable, to solve the unsolvable problems. We're going to need to leverage everyone's talents. And Sam Lewis and a number of young scientists are doing just that, and I wanted to give them a shout.
Thank you guys so much. And we've reached the end of vignette 3, so it's your final opportunity, audience, to get some questions to our panelists before we wrap up. Yeah.
I'll ask no more. So Katrina, at the very beginning, you criticized the textbook. You said boring. What would you put there instead?
All right, so for Zoom, at the beginning, you said that the textbook's examples of cells were boring. What would you put there instead?
That's a really great question. Sometimes boring is necessary. Sometimes you need to have that boring diagram, and sometimes I need to deliver a boring book report. But I think it is important that we start to break away from this kind of only showing an example of one plant cell that's like a very stereotypical plant cell, one animal cell, and one bacterial cell. I think adding into more diversity there into the organisms that we're showing could really help. And something that we've been thinking a lot about, and an idea that I'm borrowing from my PI, who is borrowing it from Scott Dawson, is that the organisms that we've picked to be these model organisms, we assume that they're kind of at the center of the bell curve for whatever phenotype, whatever trait that we're studying. But we don't know if that's true. And they could be at the extreme ends if you take like a mouse cell, if you take into account all of the different organisms that are out there that are so severely understudied, like Naegleria. So I guess I would fix it by taking out some of the nitty-gritty details and adding in just more organisms so people can appreciate that there's other cells out there that aren't an animal cell, a plant cell, and a bacterial cell. Also, a shout out to Archaea, that's also a cell.
Awesome. Do we have any more questions for our panelists? Okay, well, in that case, that concludes our panel for the evening. Thank you so much for joining us, you guys. I've had a wonderful time talking to you all about these cells. And thank you to you guys. Thank you for joining us. Look out for our next email. Our next event will be with Dr. Kristen DeAngelis, which is going to be broadly covering microbial ecology and climate change. We will be back here at the Amherst Women's Club. So we'll also, of course, be having an event virtually on Zoom again. You can check out our website at www.oebsciencecafe.org for more information. You can also check out our social media. We're on Facebook, Instagram, and Twitter. And if you haven't already done so, please fill out either our virtual or in-person sign-up sheets so we can continue to bring you guys these cafes. And with that, everybody have a great evening.